All right, so you have two nuclei. You're forming two cells. The chromosomes have completely separated at this point, and now you get the last point. Then after this is the cell itself has to split, and that occurs in the process of cytokinesis. So if you know the acronym, you can sketch out all the phases, and then we'll talk about what their specific functions are within those phases as well. All right, so what does this look like? Well, when you draw, we're going to draw them out, and then I'll show you pictures of them as well. You saw some pictures in the quiz. Interphase is not mitosis, right? So in interphase, the chromosomes are being used, and so this nucleus just looks like a big mass. Because they're all being unwound, they're being used. And then you've got this dark circle here. What's going on here? Or what is this called?
That is called a protein.
Nope. Close. The dark center in the middle of the nucleus is called?
Nucleolus.
Good, the nucleolus. But the chromosomes themselves are unwound and being used, and so they're taking up a lot of space. And so they're filling the whole nucleus, and you can't see anything, it's just a big dark blob. Okay?
Wait, which one's the big one?
Nucleolus.
So it doesn't ever spill outside the nucleus, it just stays in it.
It's just an area where the DNA is being used more than the other parts, so there's lots of enzymes packed in there as well. It's generally for making ribosomes, for making RNA.
So it just kind of cleans out the house and then unpacks everything.
Yeah, just the DNA, is just being used more frequently in that area.
Cool.
Okay, so try and use the same code. The first phase of mitosis then is prophase. Let me see. In prophase, the nuclear envelope starts to dissolve.
Sorry.
No.
And the chromosomes start to condense. And so what this looks like is you can still see the nucleus, but now you see dark and light areas within the nucleus where that unwound DNA is now being pulled into a central location, into a linear chromosome. And it's exposing the space where it was at. So you have very dark bands where the chromosome is condensing, and light bands where it no longer is. Okay? Yes?
So at this phase, are any of the outside organelles affecting, is there any change happening there too?
Yeah, so one organelle is the centrosome. And it's also preparing, getting ready to attach to the chromosomes. But for the most part, all the other organelles are just kind of getting out of the way. You know, the mitochondria is still producing ATP for whatever processes are needed for that. Everything else, I'm not exactly sure, but it's just going to stay out of the way. Yeah.
So what exactly is the time frame for this? Like, is this happening rather quickly, or does it take a while?
It depends on the cell type. So, like your skin cells are dividing daily. Other Yeah, it just depends on how fast the thing is growing. So some plants can grow really fast in a short amount of time. Yeah. All right, so that's prophase. All right, prometaphase, we don't have a nuclear envelope anymore. It's completely dissolved. Okay, so now our chromosomes are free. And our centrosomes are attaching to the chromosomes. I need another one.
So it's drawn as if there's one centrosome attaching to all of it. Is that just like a lazy way of drawing it?
So I have two centrosomes, and they're eventually going to attach to both sides.
I guess
One on each side.
Is there like one centrosome that attaches to several?
Yep.
Okay.
Yeah, so one centrosome is attaching to all of them, the other one's attaching to the other side. Yes.
They all get attached by both centrosomes?
Yeah, and we'll see that in the next phase, in metaphase. But yeah, so these are called microtubules, which are extending out of our centrosomes. And where do they attach to on the chromosome? What is that structure called where it's attached?
Protein.
Cool. So the cohesion proteins are actually between them, right? So these are your cohesion proteins. But there's another structure, and there's one on each side because it's going to attach to both sides. Do you remember what this is called? Okay, it's called the kinetochore. Okay, if you can split it up, remember kinetic energy was energy in motion. So this is the part where it attaches, which allows it to move. Okay, and it's kind of at the core. So that helps kinetochore.
So is that one word, kinetochore?
Yes. Just one word. Cohesion proteins is two. Yes,
So
Nick.
how is it attaching? What kind of a bond is that is connecting the kinetic core to the microtubule?
I'm not sure. Yeah.
Okay.
But I imagine it's probably hydrogen bonding. But yeah, I'd have to look that up. Okay, any other questions? Okay, so those are the cellular structures we learned, or the structures of the chromosome, which we learned last time. Now they are attaching to the microtubules of our centrosome. And this process is called congression. Which is the movement of the chromosomes. Eventually we're going to align them up in the middle in a specific arrangement. So congression has begun. All right, so then the next stage, metaphase. The microtubules have then guided the chromosomes so they are aligned on top of each other. And so these are homologous chromosomes, and they aren't side by side. That happens in meiosis, but they are on top of each other. And they align in an imaginary line called the the metaphase plate, right there in the middle. This is where the cell is going to divide eventually. But for now, there's nothing really there. It's just where they line up in the middle. All right, so what is the ploidy of the cell at this point? How many of each chromosome does the cell have?
Double.
2 or 1 or 3 or 4? 2, good. All right, so it's still a diploid cell. And I only bring that up because in the next step we're going to pull these chromosomes apart, right?
I have a question.
Yes.
Where did all the other organelles go while these things are lining up like this?
They're there. I don't know.
They just
We're
kinda
just ignoring them for this
shove
process.
off to the side?
Yeah.
Alright. I guess it's kind of like when a mother's a child carrying, like, organs just get shoved out of the way.
Yeah. All right, so in anaphase, what structure on the chromosome is no longer there? Kinetochore is still there. It's still attached to the microtubule. The cohesion proteins, all right, we broke them apart, dissolved them. And so now we doubled the number of chromosomes. Because it was 1 chromosome with 2 chromatids, and now it's 2 chromosomes with 1 chromatid. So what's the ploidy of the cell at this moment? We started with 2 copies of each. Now how many copies of each chromosome do we have?
Right? Or
No, it's not 1.
still 2 inside. Well,
So now we have 4.
What the freak?
It's a tetraploid at this point. Because we doubled the number of chromosomes by splitting them in half. Okay, so this one and this one are homologous chromosomes. This one and this one are homologous chromosomes. One was for the mom and one was from the dad. So now you have 4 copies of each type of chromosome. And then this one would be copy 2. Oops. Whereas before, in metaphase, you just had 1 copy of each. Or 2 copies of each. Does that make sense? Okay. All right, so there's 2 parts of anaphase. One part is the cell is elongating. And the second part, and that's just by the cell elongating, that's pulling the chromosomes apart. And then the second part is the microtubules are contracting.
Where is that mean exactly?
So those microtubules are pulling the chromosomes further to one side of the cell, on both sides, while at the same time the cell itself is elongating. So that's anaphase A and anaphase B.
And which one is which?
A is the cell elongating, B is the microtubules contracting. Okay, so the last phase of mitosis then you have nuclear envelopes reforming. You have chromosomes unwinding. And you have the nucleolus again. So it's starting to look like two new cells are forming. We're still at 4N at this point. We're still a tetraploid. And then we're ready for the last stage is, well, that is the last stage of mitosis. This is now what we need to complete for cell division to complete, right? So in animal cells, what you have are proteins which contract in on the cell membrane and cause it to kind of pinch off. And that's called the cleavage furrow. In plants, you have the addition of a cell wall. So you have a cell plate forming in between the two cells. And then once that completes, you'll then have two separate cells that are both diploid. And they'll be exact copies of the first cells. All right, so that's it. That's mitosis. We went through interphase, the cell cycle, and now we completed with cytokinesis, and now we have two identical daughter cells, and we started with one. Any questions?